Great. Well, welcome, Steve, and congratulations on the Blaisdell Distinguished Alumni Award. Your recognition for this honor is based on your longstanding contributions to our understanding of the human aging process, particularly for your discoveries at UCLA on how spontaneous molecular damage can be ameliorated by repair pathways. Just to give a little bit of background on Steve, you were a member of Pomona's class of 1970, doing a joint major in chemistry and zoology. After Pomona, you did a PhD in biochemistry and molecular biology at Harvard, followed by postdoctoral work in biochemistry at Berkeley. You've been on the faculty in the Department of Chemistry and Biochemistry at UCLA since 1978, where you are now a distinguished professor. In fact, I remember meeting you in the late 1980s when I was a graduate student at UCLA, and you were the person who actually first mentioned this beautiful school in Claremont, Pomona College, when I told you that I was thinking about being a professor at a small college. And wouldn't you know it, it all worked out just like that.
That's fantastic, Dan. I love that story. And so thank you for doing this, and to my fellow Seichens for the great recognition. And I'm particularly happy to have the name of Blaisdell associated with this. You know, a president at Pomona College for almost 2 decades, the one who kept the college small, looking at the increased enrollments through the Claremont colleges. And so I'm grateful for that. I also always think of Blaisdell because when I got to Pomona College, it really wasn't coeducational. The men were on one side, the women are on the other side. And in 1968, I was part of the invasion of the first men to go to South Campus, and I lived in Blaisdell 213. So that was actually terrific.
So do you think you could give us a brief look of your time at Pomona and how it prepared you for your career?
I'd be very glad to. Okay, so here is my Molecular Biology Institute at UCLA, and so I'm going to give you one sage chemist view of the aging process. And it all started at Pomona. And when I got to Pomona, I wasn't sure whether I was going to be in the humanities or sciences. One thing that actually influenced me greatly were these 2 beautiful, at that point, almost brand new buildings. Seaver South, which had zoology, botany, and geology, and Seaver North that had chemistry. And here are these beautiful buildings. And I only learned later that the wife of the donor who came by said, "What are these students in t-shirts and jeans doing in front of my beautiful building?" Well, okay, so here I am in 1969 in front of her beautiful building, and look, I'm wearing a shirt, although I'm sure that she wouldn't approve of the haircut. But you know, that was the great part of Pomona, wonderful people of Pomona. And what I realized was it was faculty at Pomona who were not only distinguished scholars, but distinguished teachers. And Dan is a wonderful example of that. And so in my time, I just wanted to pay a little bit of tribute to some of those professors. As a freshman, I took a field zoology course with Michael Hadfield. He took us to the Rancho Santa Botanic Gardens, and he had us waiting in the ponds looking for material until we got yelled at by the staff there. We weren't supposed to be in there. He took us down to San Felipe, Mexico. Wonderful professor. On the right is my advisor, Jack Quinlan, in chemistry. Wonderful man, later director of admissions. Jack Quinlan was the one who early as a freshman took me to what was then the Pomona Coop and for a coffee, and he asked me what my career plans were, and I told him that I wanted to be a folk singer like Bob Dylan. And he looked at me and he said, "Well, that's fine, but you know, it seems like you've done pretty well in chemistry, so maybe you want to keep chemistry as a backup." So that backup has actually worked pretty nicely for me. Great faculty in the humanities. Darcy O'Brien taught a freshman seminar my first semester at Pomona. I'm still doing his reading list for that course, Intellectual History of the US in the 1920s and 30s. Grateful for Darcy for a lifelong learning. Peter Walsh taught art history with that great mop of hair, walking in front of the slides, gave me a great appreciation for the art world. And one other faculty I have to mention is Elizabeth Cawthorn in physical education. somehow I made it past Pomona's requirements. I'm not sure what they are now, but at that point it was like 8 semesters of physical education. First semester was wrestling, which I don't know how I made it through Greco-Roman wrestling, but I did. But after that, I discovered that you could take PE on South Campus and you could take archery. And Elizabeth Cawthorn was the archery instructor. And so I took archery 6 semesters in a row. I got so good that I got my own bow. And that was sort of a little bit of a high point of my physical education at Pomona. But the professor that I really want to thank the most is Neil Cornell, professor of chemistry, who made me a biochemist. Biochemist at with totally dedicated to his students, totally dedicated to the field, and extraordinarily hardworking, and put together such a wonderful course that I still remember the laboratories, and I probably could, redo these laboratories now 50 years later. And so this is what really set me up. And also, one of the laboratories was a laboratory on gel filtration and sequencing of bovine serum albumin. And Neil Cornell pointed out that no protein was absolutely perfect. Every protein had some microheterogeneity, and it ended up, my career was studying the microheterogeneity of proteins. So
Great.
Thank you, Neil. Okay.
So Steve, I know different scientists have different views of the science behind aging. What's your big picture of how we lose function as we grow old? That's a question that's on my mind all the time.
Yes, and mine as well. Well, I love this. Chemists study reactions, all right? So, A goes to B. So on the left, you see me as a freshman at Pomona College, and on the right, you see me more recently. And so this is a chemical transformation, okay? You take me, at 16 and me at 70, you put 54 years of incubation in some sort of a chemical pot at 37 degrees, and you get that. And my only question is, what happened? What happened? What happened to that young man there? And, as a biochemist, we're taught to appreciate the beauty of molecules, the beauty of molecules of DNA, RNA, protein, small molecules, and crafted perfectly. All the bonds are in the right place. But what we learn by leaving things out on the bench overnight and having things degrade is that time is an enemy, and that with time, these molecules are on their way to less functional molecules. And in fact, at the end, it's all going to be ammonia, water, and carbon dioxide, molecules that don't have a lot of function. So that's the problem. And so here are some of these, structures where you have something with just 6 carbons, glucose on the lower left, a protein with maybe 2,000 carbons, chromosome with DNA, maybe 1 billion carbons, and all of those bonds have to be perfect. And so how do you do that? And you don't. So there's degradation. And so I think, in looking at the physiology of aging, I love this Gary Larson cartoon, because here are the 2 dogs on the porch, and not only are they not going after the male carrier, but the male carrier is a cat. All right, so something's happened. And so the question is, what happened to me in those 54 years, and what happened to them dogs? Okay, and if you look, it's not one thing. It's a whole bunch. It's a laundry list of problems coming down. Replacement of muscle by fat, I'm there. Okay, loss of bone density, yeah, that's me. Skin loses elasticity, yeah. Okay, hair loses pigmentation, I guess that happened. Coming all the way down to, loss of neurons and memory losses, okay? So it's everything going on, and presumably everything's going on because all the molecules that make those normal functions are now not quite having the same bonding. So, one can look at, aging in terms of sort of totally honest truth. And so, here is singer-songwriter Chris Christopherson, class of '59, great song, Shipwrecked in the '80s. And you're shooting through space on this river of time you're riding, and it's whirling and sucking you deeper down every day. Okay, that's the aging process. Okay, so that's the totally honest view. All right, now what perhaps maybe a too honest view. And I love this picture of Muscle Beach and the gymnastics people because I know you're all looking at that lovely specimen of humanity flying in the air there, this wonderful young woman. But I don't want you looking at her. I want you looking at those two old farts in the foreground, because they probably were in the air just a few minutes before. They're just not as photogenic. So the fact of the matter is, in spite of this chemical instability, we do pretty well, okay? And we do pretty well to last, up to a century or perhaps even a little more. So how does that occur? And so, the question is, how do you fight off that spontaneous chemical damage? Now, for DNA, where any one bond breaking can lead to a mutation, we know how we fight that off. And of our 19,000 human genes, about 400 of those encode proteins that basically take care of DNA. And you could imagine that these 400 proteins are like these workmen here, going up and down the DNA strand, looking for damage, looking for chemical damage. And when they find that chemical damage, they fix it, right? So that was very well known for DNA, but was not known for proteins and small molecules when we started. And, I've always been interested in proteins. And in proteins, lots can go wrong as well. And it turns out that, for a protein, everything that's sort of good for us is bad for us. So oxygen, toxic species for protein, although, the brain can't do without it for more than about 6 minutes. Glucose we can't do without, but glucose is chemically reactive, beats up proteins. Water itself, okay, leads to hydrolysis of proteins. And so this kind of attack, magnified with time, leads to the degradation. And so I like to look at aging as basically warfare. And it's warfare between chemistry and biology. So unfortunately, Dan, the chemists get the bad chemistry, because that's the spontaneous damage at multiple sites. And I once had a colleague say, look, Steve, chemists take such a beating anyway. Okay, can't you blame this on the physicists? Isn't this entropy? All right, so maybe this should be bad physics. All right, but that is the damage. But biology fights it. And biology fights it because we can recognize damage, and we can actually, in proteins, we can actually remove them, chew them up and make new ones. But what we learned is we can also repair them. And these repair pathways then are what I've spent my career doing. And so we now have repair pathways for damage of molecular oxygen in methionine sulfoxide, repair pathways for glucose damage, and in work in our laboratory looking just as water as a toxin, the damage to protein aspartyl and asparaginal residues.
That's interesting, Steve. I look at proteins and they're structurally much more diverse, much more complex than DNA duplexes. I can only imagine that the repair mechanisms are a bit more complicated. And I was wondering if you could just share a brief view of what you mean by protein repair and how that works in a protein.
Sure. So, what we have focused on is one part of protein damage. And proteins are made of linear chains of 20 amino acids. And what we've learned is that evolution screwed up, okay? Evolution screwed up because two of those amino acids probably shouldn't have been there, okay? And they shouldn't have been there because they're very sensitive to spontaneous damage. And so we've looked at the damage of these two amino acid residues, and then we've looked at their repair or their recognition for proteolysis. And so the idea then is, if you can fix the damage or get rid of the damage before it affects the physiology, you'll have a nice health span. If you don't, you're going to have problems. And so the two amino acids that I believe shouldn't have been there are asparagine and aspartyl. And so for the chemists in the audience, you can look at the structures in green. That's what's good. Okay, what's bad about these guys is that, in fact, they go through spontaneous chemistry, and this chemistry makes all of those 5 bad components shown at the bottom. These ring forms that are in the L stereoisomer, the D stereoisomer, and in ones that we have just isomerized aspartate itself. And what we learned, though, was that the major form of this damage, actually, like DNA, can be recognized by a protein. And there are proteins that go and they check out other proteins. They check out the aspartic acid residues. And if they see an aspartic acid residue, that's fine. They leave it alone. If not, they go through repair pathways that I've shown here that involve the transfer of methyl groups. And that transfer of the methyl group can lead to the repair. And I love, Dan, to show this with my arms, okay? So for non-chemists, okay, this is a polypeptide chain. That's the long chain. Here's a side chain, my hand, of an aspartyl residue. The problem with these residues is that the side chain's reactive. Those are my fingers, okay? And that's a reactive electrophile. In the main chain of the protein, there's a nitrogen that's not a too bad nucleophile. Nucleophile, electrophile, plus likes minus, get together, and we have this succinimide form. That wasn't supposed to be there. And in fact, that succinimide form can hydrolyze spontaneously and actually reroute the polypeptide chain so it's no longer linear. There's now a kink in it. And so as we sit here, our proteins are being kinked. Now, the good thing is this repair methyltransferase that we found comes along and actually can look at this free carboxyl, my thumb here, methylate it, allow the ring form to come together and give us a chance to come back to the normal configuration. And so basically we have a repair enzyme, which is a dekinking enzyme. Now, when we found this enzyme, we said, great, okay, so this enzyme, is presumably necessary to keep proteins alive through aging. And so we went on a very long journey that's continuing of what happens if we mess with the gene? What happens if we have an organism where you don't have this repair, and does that lead to a short half-life? And alternatively, maybe there's not enough of it. Can we put a little more in genetically and maybe give a long half-life? And these experiments have gone on. It's more complex than I expected, but there are a few results that I do want to share with you. One from Philippe Grepin's lab in France, who looked at seed aging. Now, we talk about aging in humans. Plants have aging too, and a major form of plant aging is in their seeds, because a seed is basically equivalent to a human fetus, maybe about 6 months, that's then wrapped up in a hard shell and thrown out into a miserable environment where that seed might have to survive in that environment for maybe weeks, months, maybe years before it can germinate and that embryo can grow. And so what the Grepin lab showed was, in fact, seed survival, okay, is enhanced if you bring more repair methyltransferase, and if you take away methyltransferase, the seeds don't last as long. So it is important for survival there. And in experiments done by my colleague Claire O'Connor at Boston College, she looked at fruit flies where you could manipulate the level of this enzyme. At 25 degrees, a normal temperature for a fruit fly didn't matter in terms of the lifespan. The lifespan was the same with or without the overexpression of this repair enzyme. But when you stress these flies, you put them at 29 degrees where they're hotter, more chemical reactions. Now the flies with the overexpressed enzyme live significantly longer, so that this actually goes on. Experiments in our lab with soil nematodes, we can also show that the soil nematodes have longer lives with overexpression. Now, in the soil nematode, it means going from 15 days to 25 days, so it's not quite living 150 years. What happens in humans and other mammals? We don't know yet, but we'd like to know. We'd like to know if this might work.
Steve, can you give us a brief update on your recent work with Alzheimer's disease? I mean, this is an area of critical unmet need. Any thoughts you might have on possible therapies for those affected by this disease?
Absolutely. It's a grim story, Dan. It's a grim story because I tell people sometimes that I can be an Alzheimer's, physician, because I can tell, someone's loved ones that there's nothing we can do as well as anyone else. So right now, really no therapies in spite of massive investment. And so we had studied Alzheimer's disease back in the 1990s. And when we studied Alzheimer's disease, because Alzheimer's disease is not a new car defect, okay? No one gets Alzheimer's disease as a child. No one gets as a teenager. even with the worst predisposing mutations, okay, I think the earliest cases are after like 38 years. And fortunately for most people, it's, late 70s, late 80s, 90s, okay? So it takes a long time for this disease to show up. And we thought, okay, this disease is one of the hallmarks of this is extracellular plaque that's made of the beta amyloid protein, and that protein has aspartic acid residues. Maybe the damage to those residues is what's leading to Alzheimer's disease. And we actually tested that, and the hallmarks of Alzheimer's disease pathologically are two. And one of them is what's shown in the center of this slide, this extracellular plaque. And this plaque is basically an aggregate of the beta amyloid peptide. And I'll talk more about that later. Okay. The other hallmark is inside a neuron, which are neurofibrillary tangles that are made by aggregated tau protein. And the aggregated tau protein can lead to the death of a neuron. And people in the field have fought over the last couple of decades of which is more important, the neurofibrillary tangles from the tau protein or the plaque from the beta amyloid protein, or BAP. And so it's been described as a contest between the Taoists and the Baptists, okay? And let me tell you that today it seems very clear that the Taoists have won. And it's a painful victory because so much work went into looking at the plaque. And so the plaque turns out to be the development of plaque does not correlate with Alzheimer's disease. And in fact, therapies have been done that can remove the plaque. You can remove the plaque, you don't affect Alzheimer's disease. So the plaque looks like burned out extracellular matter. And okay, so we found isoaspartyl and other damaged residues there, so what? All right, so then how do we go back to what's really killing the brain? And it's really looks like the neurofibrillary tangles that come from the tau protein, all right? But we know that mutations in the beta amyloid peptide can lead to early Alzheimer's. So a new model has now come, which is really a paradigm shift, which is that the beta amyloid may not be what's on the outside in plaque. It may be the material inside the cells that then can aggregate and then cross-seed the polymerization of the tau protein. Tau protein is really bad because when it polymerizes and aggregates, it only kills that neuron, but that neuron then can kill the next neuron over. And that's where you have in Alzheimer's disease, the spreading of disease, where you start out with memory defects in the hippocampus, and you end up with those pictures I showed you where half the brain is actually gone from the loss of neurons. So we then started looking at this again, and we said, okay, what intracellular form of beta amyloid might give rise to such cross-seeding? And so what I've shown here is the 42 residues of this beta amyloid peptide. And the region that's shown highlighted in purple is a region that's responsible for the aggregation. And what we noticed was in that region was an aspartyl residue at position 23. And that residue was right next to a number of other residues that when they're mutated cause early Alzheimer's disease. So we said, aha, could the isomerization of that position be affecting the aggregation of this molecule? And so wonderful graduate student, Rebecca Wormack did this with fantastic structural biology colleagues at UCLA, and we got a three-dimensional structure. And the crucial part of that three-dimensional structure is shown at the lower right. And what it shows is that the isoaspartic acid residue, that side chain where you've now done a kink, leads to a new type of structure, a new type of stickiness, and leads to, in fact, enhanced stickiness of the beta amyloid protein. And not only stickiness, on the left, I've shown you a region of the tau protein. Now, this is the bad guy, remember? And so there's a similarity between these structures, and we're wondering now whether, in fact, it's this isoaspartyl form of the beta amyloid protein inside that can seed the tau protein and cause disease. Okay, so where does that leave us? Okay, leaves us with a pickle, and this is the pickle, is that the repair process I've talked about can actually repair intracellular beta amyloid and do a pretty good job of it, okay? But after 60, 70, 80, 90 years, all that has to happen is one isospart has to slip by long enough to be able to form an aggregate and seed tau. And once that happens, you're down a pathway. So what's the situation then for Alzheimer's disease? Okay, well, we can certainly try to enhance these pathways, okay, and try to keep that from happening, but there's probably 50 different ways that tau protein can be seeded. And so it's like a roof with 50 holes in it. I can patch up the hole that's due to isospartal. The other ones may be tougher. And what it says is that maybe we shouldn't be focusing then on the amyloid, the beta amyloid, because that's going to happen in spite of all the repair, and to try to stop things later on with tau. So that's where we are now. It's a little disappointing, but I think at least now we know where to go. And there's been so much effort by very good people, so much money spent, but looking at extracellular beta amyloid probably wasn't the thing to do. But now we can turn the ship around and hopefully go in the right direction.
Great. So having known people that work in your lab, I know that you're a very on-site professor. A lot of university professors may do research at a distance through their graduate students, through their postdocs, but I know Steve Clarke is always in lab, always urging students on, giving them lots of encouragement, but also I think most people would describe you as an eternal optimist, always thinking that things are going to work. And that brings me to my final question, because it seems to me you need optimism to be working in this area. And what makes you optimistic that possibilities for slowing the ravages of the aging process? What's going to be the story? How's it going to turn out?
Yeah. Well, Dan, you're absolutely right about my optimism in spite of everything. One of my colleagues calls me Dr. Pangloss from Indeed, which I don't think is a compliment. So, basically, what we're after is, enhancing human health span. And, hopefully that, the best type of life is, going strong till 95 and then dropping dead, one day in your garden. Okay. So now, is that going to be easy? And probably not as easy as overexpressing just one protein repair gene in humans. Okay. I think we're not naive enough to think that, but what about sort of conditions that might actually enhance the activity of many protein repair genes? And so, and other genes involved in looking at damaged biomolecules. So can we learn how to enhance the activity of these enzymes? And are there some tricks that might actually increase the activity of repair degradation systems and slow the physiological loss of function in aging? So that's basically where we're going. And so, I go to lots of aging conferences, and at these aging conferences, somebody will come up with their new, anti-aging remedy, whether it's resveratrol or whatever. And at the end of the meeting, someone will stand up, sometimes it's me, sometimes somebody else, and saying, listen, how is this any better than what we already know? And what we already know is basically mom's advice, okay? Okay, so good diet, exercise, sleep. There's something chemically magical about the three of those. Good diet, we're beginning to understand. What's so magic about vegetables? Okay, are they sort of bringing their defense against oxidant damage from plants, where they first had to deal with oxidant damage, to us? The benefits of things like coffee, which I think is now on the good side, omega-3 unsaturated fatty acids, but how these dietary components might actually affect these enzyme activities. What happens in exercise? What happens in muscle, where you get these micro tears that start processes of muscle building and health? And the importance of sleep is basically a repair process. I think the more we learn about sleep, the importance of taking some time off and trying to fix some of that damage why things aren't working. And so, what keeps us going is thinking that these things may work, at least in part, by enhancing molecular repair. And so my final optimism is, here the fountain of youth, okay? And so here I am sitting on the left, okay? And, this pool, which I might imagine is some activating broth for molecular repair, maybe some combination of all the good vegetables, right? And then coming out on the other side doing better. So that's my optimism. And, just to get back to Jack Quinlan and my desire to be a folk singer, this lyrics by Bob Dylan, I think really sums it up for me. May your hands always be busy. May your feet always be swift. May you have a firm foundation when the winds are changing shift. May your heart always be joyful. May your song always be sung. And may you stay with repair enzymes forever young. So that's my optimism. Thank you, Dan.
Great. Thanks, Steve. This has been a real fun conversation. And I really can't tell you enough. Meeting a Pomona grad when I was a graduate student and finding out just how proud you were of the school and what it did for you, and that places like this exist, it was just eye-opening to me. And then to really learn that you as a Pomona grad, the department, seeing you at UCLA, winning teaching awards, doing research at the highest level, the department's so proud. The biology department is proud. The school is very proud, clearly. And you've been a huge supporter of our department. You've participated in reviews. You've urged us on to reach higher in the things we do with research and teaching. And you're just one of my most favorite sagehens out there. So I'm really happy that I had a chance to participate in this interview, and just want you to know how special you've been to me as a professor at Pomona.
Well, thank you, Dan. And right back at you, okay? My daughter graduated from Pomona as a chemistry major in 2012, and I think she spent many hours in your office where you're basically patiently and wonderfully explaining organic chemistry to her. So thank you for all you've done for Pomona and your colleagues.
No, we really appreciate it. And this award is just richly deserved all the way around. And I'm really happy that you're being recognized in this way. It's spot on.
Thanks so much.
Thank you, Steve.